Hello, so my name is Ken. In my project, I discussed how we can retain CHD7, an epigenetic regulator, in the nucleus to combat breast cancer metastasis. Breast cancer is the second leading cause of cancerous death for women around the world, and about 12.4% of women are diagnosed with this disease at some point during their life. The primary reason it's so deadly is because of metastasis, and this is what happens when some cancer cells from the primary tumor invade into the nearby tissue and intravasate into the circulatory system where it can spread and form secondary cancers. In fact, this accounts for about 90% of deaths from breast cancer. My study addresses the fundamental question of how cancer cells in the primary breast cancer tumor become metastatic and spread to other parts of the body. The gene I looked at was CHD7, which encodes an epigenetic regulator. Recent studies have revealed epigenetic regulation as a critical mechanism for breast cancer pathogenesis and metastasis. CHD7 is an ATP-dependent nucleosome remodeling factor, which means that it acts in the nucleus to regulate the density and localization of nucleosomes. Mutations in the CHD7 gene account for about 70% of cases of CHARGE syndrome. However, the potential function of CHD7 in breast cancer metastasis has never been studied before. First, I wanted to determine whether CHD7 is expressed in breast cancer cell lines, and if so, where it is localized. To do this, I performed immunostaining on 3 different breast cancer cell lines. The first column you see is the blue DAPI stain nucleus. The second column you see is the green CHD7 stain. And what I found was that all 3 breast cancer cell lines expressed CHD7. However, the localizations of their CHD7 differed. In MCF7, which is my least metastatic breast cancer cell line, the most amount of CHD7 was localized in the nucleus at about 90%. However, in my medium metastatic breast cancer cell line, 468, only 25% of the CHD7 was localized in the nucleus. And finally, in my highest metastatic breast cancer cell line, 231, the least amount of CHD7 was localized in the nucleus at only 5%. So now that I found this result in a breast cancer cell line, I wanted to see if it also held true in invasive breast cancer tissue from patients. To do this, I obtained invasive breast cancer tissue from a patient and performed HE staining on the normal and cancerous areas. Then I performed immunostaining on adjacent sections. Once again, the first column you see is the blue DAPI stain nucleus. The second column you see is the red CHD7 stain. And the green you see here is CDH1, which is E. cadherin, and it is an epithelial marker. What I found is that in the normal breast area, over 99% of the cells had their CHD7 localized in solely the nucleus. However, in the cancerous breast area, over 30% of the cells had their CHD7 localized in both the cytoplasm and the nucleus, and some of these cells even had their CHD7 localized in solely the cytoplasm. These results clearly support the clinical relevance of the subcellular localization of CHD7 in breast cancer. So far, I've showed that in normal epithelial cells or non-metastatic breast cancer cells, the CHD7 is localized in the nucleus where it can function. However, in the metastatic breast cancer cells, the CHD7 is localized in the cytoplasm, unable to function. These results suggest that CHD7 may regulate breast cancer metastasis. And to target this large issue, I first tackled the smaller question of whether or not CHD7 regulates breast cancer cell invasiveness. To do this, I knocked down CHD7 in 468 cells using 2 independent siRNAs. I confirmed that the knockdowns were successful by performing a Western blot analysis and then performed a cell invasion assay. This shows my setup, and these are images of the 468 cells that invaded through the filter. What I found is that in the 468 cells with their CHD7 knockdown, there's a significant increase in the amount of invasion that occurred compared to my controls. This shows that knocking down CHD7 increases breast cancer cell invasiveness, in turn suggesting that endogenous CHD7 can actually inhibit breast cancer cell invasiveness. With this in mind, as well as the fact that in metastatic breast cancer cells, the CHD7 is localized in the cytoplasm, I next addressed the question of how CHD7 is exported out of the nucleus into the cytoplasm of metastatic breast cancer cells. I used a computer program to map out 2 potential nuclear export signal sequences, which I labeled NES1 and NES2. Then, to test their functionality, I performed a reporter analysis. When the NLS-GFP construct was fused with NES1, large portions of the GFP was exported into the cytoplasm, suggesting that NES1 is functional. However, when the NLS-GFP construct was fused with NES2, large portions of the GFP was still localized in the nucleus, suggesting that NES2 is not a functional nuclear export signal. So now that I found NES1's functionality through a reporter analysis, I wanted to see if it was truly functional in endogenous CHD7. And to do this, I mutated the NES1 sequence of endogenous CHD7 in 231 cells using the CRISPR-Cas9 technique. Panel A shows the general principles behind it, and in panel B shows the specifics of what I did. I generated a plasmid expressing Cas9 fused with GFP, as well as guidance RNA with 20 nucleotides targeting the NES1 sequence of CHD7. Then I co-transfected 231 cells with this plasmid, as well as donor oligo, which contains a mutation of leucine into alanine and a PSC1 site. Through single cell cloning, I obtained 15 independent cell lines. Then I performed PCR analysis and identified one heterozygote and one homozygous cell line. And these results were then confirmed through sequencing. So now that I've acquired my desired homozygote mutant breast cancer cell line, I examined the localization of its mutant CHD7 compared to the wild type. What I found after performing immunostaining was that in the wild type 231 cells, only 5% of the CHD7 is localized in the nucleus, as consistent with earlier. However, in my homozygous mutant 231 cells, over 60% of the CHD7 is now localized in the nucleus, which suggests that disrupting the NES1 sequence truly leads to increased nuclear localization of CHD7 in highly metastatic breast cancer cells. So now the question remains of whether or not this result has any biological significance. And to test this, I performed a cell invasion assay. So what I found is that compared to the wild type 231 cells, in the mutant 231 cells, the invasiveness of these cells was reduced to 30% of wild type invasion levels. This shows that retaining CHD7 in the nucleus through mutating its NES1 sequence significantly reduces breast cancer cell invasiveness. So far, I performed all of this functional analysis of CHD7. Next, I wanted to determine the mechanism by which CHD7 operates to actually inhibit breast cancer invasiveness. And to do this, I first performed reverse transcription PCR to determine CDH1 expression in wild type and mutant 231 cells. CDH1 was detected in the mutant 231 cells, unlike in the wild type 231 cells. As I said earlier, CDH1 is E-cadherin, an epithelial marker, and it also reduces cancer cell invasiveness. With this in mind, as well as the fact that CHD7 is a nucleosome remodeling factor, in panel B, I developed my working hypothesis. In the wild type 231 cells, the promoter region of CDH1 is densely packed with nucleosomes, preventing its transcription. However, in my mutant 231 cells, I've retained CHD7 in the nucleus so they can act upon the promoter region of CDH1, loosen up its nucleosomes, and allow for CDH1 transcription. To test this model, I performed chromatin immunoprecipitation PCR. First, I used anti-CHD7 antibody and chip, and found that, as expected from my hypothesis, in the mutant 231 cells, CHD7 is associated with the promoter region of CDH1. Next, I used anti-histone H3 antibody and chip. Once again, as expected from my hypothesis, in the mutant 231 cells, there was reduced histone associated with the promoter region of CDH1 compared to the wild type 231 cells. These results support the idea that CHD7 epigenetically upregulates the expression of CDH1 in mutant 231 cells. So now I drew a model of what I believe to be happening in the different cell types. In the normal epithelial cells, or non-metastatic breast cancer cells, the CHD7 is in the nucleus, acting upon the nucleosomes as it should, and metastasis is inhibited. However, in the metastatic breast cancer cells, the CHD7 is exported into the cytoplasm through its nuclear export signal, unable to function, therefore metastasis occurs. If I can retain the CHD7 in the nucleus of these metastatic breast cancer cells, it can once again act upon the nucleosomes as it should to inhibit breast cancer metastasis through the promoter region of CDH1 or any other gene. So in conclusion, in the epithelial cells of invasive breast cancer tumors, large portions of CHD7 is localized in the cytoplasm, unlike its nuclear localization in normal duct epithelial cells. I identified an NES sequence critical for exporting CHD7 into the cytoplasm of these cells and found that mutating the NES sequence leads to an accumulation of CHD7 in the nucleus of metastatic breast cancer cells and a significant reduction in their invasiveness. Now, while in the nucleus of metastatic breast cancer cells, CHD7 actually reduces the nucleosome density of the CDH1 promoter and promotes CDH1 transcription. This study helps address one of the most urgent and challenging questions in the field of breast cancer research. How does a breast cancer cell in the primary tumor become metastatic? Our results collectively suggest that retaining CHD7 in the nucleus may serve as an effective therapeutic strategy to inhibit breast cancer cells from becoming metastatic. So in the future, I'd like to identify the signaling pathway that regulates NES activity in CHD7, as this pathway may serve as a potential drug target. Next, I'd like to identify other genes that CHE7 regulates to inhibit breast cancer metastasis or invasiveness through techniques such as RNA sequencing. And finally, I'd like to test the effects of mutating NES1 sequence of CHE7 on metastasis using in vivo experiments such as xenotransplantation. And last, I'd like to thank my mentor, Dr. Lizhong Wang, as well as the rest of his lab and UAB for supporting me throughout my project. Thank you for listening.